All right, let's just get started. So as you can see, this is the BIOME Summer 2021 Stanford webinar series, and we're super excited to have you all here. This is such a great group of people, and we're incredibly lucky to have this much enthusiasm, for biology and synthetic biology and bioengineering, and you all had some great responses. And yeah, we can't wait to do this with you all. Now, because there are a lot of you, let's see, what's the best way to go about doing this? I guess Colin and I can do introductions out loud, but then everyone else can just do like a mini blurb in the chat, if that's all right. So hi everyone. I guess Zoom is easy because you can see my name, but my name is Alan, and I am a junior at Stanford University studying biochemistry. And Colin, do you want to give a short intro?
Yeah, I'm Colin. I don't know if you can see my face right now, but that's okay. You saw as you're coming in. I'm going to be a senior, and I study bioengineering at Stanford. I'm super excited to work with you all. And Alan will be leading the first couple. You'll see me on the next couple, and then Alan will finish off the end. So be good.
Awesome. So there are quite a bit of you, and so please, use the chat. I strongly encourage, you all to be engaged in this, and to make the most of this time, because we only have an hour. And so, use the chat, say hi to people, introduce yourself, maybe where you're from, what state, because I think we have people, across the country, maybe even internationally. So super exciting group. Favorite animal, yeah, that as well. So name, grade, where you're from, and favorite animal. And while you're all doing that in the chat, I can, proceed and give you some details about how this whole thing will work. And so this is a webinar series, and we do have a YouTube channel. And so it's called Stanford Biome. Feel free to check us out and subscribe. And there's going to be a total of 9 webinars. And so, all of you today, of course, is June 25th. And the webinars will last until August 20th. And so there's going to be 9 total, same time every week. And the last session, which is on August 27th, will be reserved for final presentations. I believe we alluded to this in sort of the registration process, but how it'll work is you'll be in a group of 3, and there will be like a mini presentation. And this is no pressure, no stress. We're not grading you or anything. This is just so that you can apply what you've learned into something you're actually really interested inside. Now, I know that you all probably don't know each other yet, and so Colin and I have decided that we're going to group you all into groups of 3 depending on interests. And we'll deal with that later on, so just check your email for more info in the future. The format is going to be 5 minutes of the presentation with about 1 minute of Q&A. And because we have such a big group, this is going to be a little bit strict. We can go a little bit over, but hopefully not too much. And we really don't want to limit you all. So, feel free to make a PowerPoint or Google Slides, make it festive, make it scientific, make a video, make a interpretive dance, whatever you want. But this is up to you and group of 3. And yeah, we will give you more info as the weeks go by. But keep in the back of your mind as you're learning all this stuff, remember to think about what you're interested in and what you want to learn about more during the presentation. Also, be engaged, right? So use the chat. Because there are 25 of you, I can only see a few in the front. So if you want to say something, raise your hand, then I think you'll pop up to the front and I'll be able to see you. So raise your hand, use the chat, and I'll also be using Poll Everywhere. Colin might have, different engagement activities, but, some of you might have heard about Poll Everywhere. It's really easy to use. It's just like a website and you type in your answers there. So I'll be using that. And turn your videos on if it is okay. I realize that people are Zoom fatigued. Colin and I were students as well, so we completely understand. We're not that much older than you all. So, however, I would appreciate if you turn your videos on if it's reasonable. That way, we can build a little community here. It's not just talking to some names on the screen. Be respectful, of course. You're all high schoolers, so I don't need to tell you all more about that, but be respectful. And have fun learning. So these are, some of the details regarding the next 10 weeks. Are there any questions? And Colin, I see the chat blew up, so it's popping in there. Are there any important questions in there? We're good? All right, awesome. And here's a schedule of the topics we're going to be covering. A is me, Alan, and C is for Colin. And so today, as you can see, we're going to be going over an introduction to DNA. And because we have a diverse group of people, some are freshmen in high school, some are seniors in high school, and there's a range of biology experience, I don't want anyone to feel left out. And so some of this might be repetitive and some of this might be, stuff you already know. But I do hope that we all learn something out of these beginning sessions. And hopefully, as we go along, everyone will learn a little bit of synthetic biology. Awesome. And of course, last day is, the spotlight's on you all. Final case study presentations. Okay. If there are no more questions, I'm going to do a poll everywhere experiment. Let's see if this works, right? So how poll everywhere works is there's a link at the top, pollev.com/AlanTong472. You should be able to put that directly in the URL. And this question will pop up. What excites you about bioengineering? And you should be able to enter a response. If you can't, definitely let me know and we'll try to figure it out. But yes, let's see if I get some responses popping in.
Maybe while you see the responses, you can tell you, or Alan can share what excites him about bioengineering. Want to share, Alan?
Oh, me?
Yeah.
Oh, yeah, sure. So a lot came all at once. But I guess for me personally, I'm more on the biochemistry side. And so I enjoy things like enzymology and bioengineering on sort of the chemical scale, modifying the molecules to really manipulate biology. Now, see, manipulate sounds like a scary word, but that is what we're doing, right? We're manipulating biology. How about you, Colin?
Yeah, I think that's awesome. I like the tool building aspect behind it. I think just as much as computers have been really useful for the last, 50 years, I think in the future, a lot of industries are going to be replaced by synthetic biology. And so I think that's really, really the future of things. So I like that. I like the tool building kind of behind it, i.e. like CRISPR as an example.
Yeah, I see CRISPR in here. Oh gosh, it's really changing all the time. But I saw CRISPR somewhere. Where's CRISPR?
Below cancer.
Well, okay, yes, right there, CRISPR. Yep, right there. Right, so I see genetics, creativity, DNA, code, and this is actually a really good, this might seem out of place, but this person who entered this, it might mean like computational, biocomputation. It could also mean like, the code of biology, right? How do we understand what makes us us?
I like me. That's funny.
I like me as well. Me, period. Cure, right? We can solve a lot of, diseases. People are even saying, what if in the future aging is just a disease that you can cure, right? That's something that you can think about, right? Medicine, definitely. Editing, technology. Yeah, all these are really good answers. Okay, so I'm glad poll everywhere works. If it did not work for you, please let us know. And we'll try to figure things out. I'll leave a smiley face too. That's a good one. All right. Let's move on. Now, why is the slide not letting me continue? Okay, there we go. So as I mentioned in the very beginning of this webinar, today's focus is going to be on DNA. Now, I'm sure that every single one of you in the audience have heard about DNA, at least in some form or another, right? You might have studied it in detail in an AP biology class, for example, or you might have heard it just on passing, right? It's in our DNA, or it's even made it into hip hop, Kendrick Lamar's DNA song. So DNA, DNA is everywhere. It's a really iconic term, and today I really want to dive into what it is, how we discovered it, and why it's useful, right? Why is it interesting? And so of course we have the obvious question: what is DNA? Right? What is it? You hear all the time, but what is it? Let's get some responses. It should be the same link, I believe. Alan Tong 472, right? Exactly. All right, we already got one up. Deoxyribonucleic acid, right? Oof, vocab. We'll discuss what that means.
That's an impressive spelling that quick. I don't think I could spell it like that.
Yeah, yeah. We got the code of life, right? There's that word code again, right? So it's just like a computer, right? Biology might be programmable, codable. Blueprint, right? Coding of the body. It's fundamental. Genetic material. I like that. I like that. Right? So we're going to be talking about genes a lot. Genetic Legos. I like that term as well. People often say that our cells are kind of like Legos. DNA is also kind of like a Lego as well. You have different pieces and you piece them together and you get some information out that tells us how to make us. Unique molecular coding, twisted ladder, right? So now we're getting into the shape, right? What does it look like? I'm sorry, it's been a bad day. I hope you feel better. Blueprints, right? So we'll get into central dogma for sure. What makes us us? BTS. Oh, is there a BTS song called DNA? Is that what that's from? Let me see the chat.
There's a BTS meal. That's all I know.
Mm-hmm.
There is one. There is a BTS song called
DNA. I see.
Thanks, Ashley.
Building block, blueprint. Right. Exactly. Okay, so here's another vocab term coming up. So nucleotides. Right, so more structure, more chemistry, right? Phosphate, sugar, nitrogenous base. And for those of you who might not have heard about the chemistry behind DNA before, we'll get into all this in just a bit. But I think what's really good about all these responses is, both the generality and the details of it. Because on the big picture wise, it's important to realize that DNA is the code of life. It makes us who we are. But if you get into the nitty gritty, right, what is it? It's a chemical, right? Just like water, H2O, just like the graphite in your pencil lead, the carbon in your pencil lead. DNA is just a chemical. It's a certain arrangement of atoms bonded together that just has this amazing function that gives life all these amazing properties. So we got, a slew of answers, and these are all really good answers. I'm not sure why it's not letting me go forward. Pull over, it might be a little bit sassy. But here we go. This is like the very first response that we got, right? DNA stands for deoxyribonucleic acid, right? Now that's a big mouthful. Pretty much no one ever says deoxyribonucleic acid. Everyone just says DNA, right? It's just shorter, it's simpler, it's nice and sweet. If you ever want to, brag in a party, just bring it up, deoxyribonucleic acid. But that's what DNA stands for, right? DNA. And it's called a nucleic acid. Anyone know why it's called a nucleic acid? I'll monitor the chat as well. Why is it called a nucleic acid? Any guesses? Exactly, right? Rohan got it. It's in the nucleus. All cells, I shouldn't say all cells, our cells, all of our cells, not all of our cells either, because our red blood cells don't have. Anyways, as you can see, biology has all kinds of exceptions, but a lot of cells have what's called a nucleus. It's kind of like the core of the cell, and in that nucleus contains our DNA. That's why it's called nucleic. It's in the cell's nucleus. Why is it called acid? Anyone know? I think Rohan's on getting there, but why do we call it an acid? So someone said H bonds, hydrogen bonds. Other guesses? What does it mean for something to be acidic? Acid. H donor. It's got acidic properties, pH less than 7. Exactly. Right. And so the idea is that if you were to taste DNA, theoretically, it would taste sour. Because acidic things taste sour, right? They release what are called protons, H+. I saw a video on YouTube that said that we can taste protons, they taste sour. So DNA is indeed an acid. And some people are getting into the chemical details. It's actually the phosphate group that's acidic. The phosphate can release protons, making DNA acidic. And we'll get into the structure later on. But for now, this is what DNA stands for. Now, here's a question that's been plaguing, and I mean plaguing people for literally centuries. Why do we look like our parents? Right? What's the mechanism of heredity? Because if you look around, right, hair color, eye color, skin color, right, it's pretty similar, right? If you have your biological parents, chances are you're going to look like, one of them or both of them or a mixture of some of them, right? But it's also not simple as well, right? What's the mechanism of heredity? And of course, it's not just limited to humans, right? Dogs as well. They look really similar to their parents. All organisms pretty much, right? So peacocks, giraffes, whales, bacteria as well, right? Archaea. So this is something that's central to all of life that we know of, right? Life and organisms, they look like their parents. So people thought, well, something from the parents must be passed down to the offspring. Of course, now we know it's DNA, but I want to get into the story of how this worked. And here was one of the really first experiments that showed about the passing of traits. So here's the idea. Some of you might have seen this experiment before, but there was this dude called Griffith. I don't want to get into the nitty-gritty details of this experiment, but here's the idea. There were two types of bacteria, S and R. The living S strain, the S bacteria, killed the mice. So literally, if you injected the mice with the S bacteria, they would die. If you inject the bacteria into the mice and they were R, the mouse would live. Now, here's where, this experimenter tried to get a little fancy, right? What if I killed the S bacteria? Remember, the S bacteria are the bad ones. They're the evil ones, right? They kill the mice. What if we heat up the bacteria? We cook them. We heat shock them. And after we cook them, now they're all dead, right? Bacteria can't survive that high temperature. What if we inject the dead bacteria into the mouse? Guess what? It lives. Now, that shouldn't be surprising, right? Dead bacteria can't really do anything because they're dead. And so they can't release the toxins and they can't kill the mice. Here's where the breakthrough happened, okay? What if you mix together, and again, this was kind of serendipity too. Like, how did he know to do this? Just for fun, right? Let's see what happens. If we mix together this group and this group, right? So the living R strain and the dead S strain, you would predict that the mice would live, right? Because both these arrows point to living mice. The living R strain would allow the mice to live. The dead S strain allowed the mice to live. Combine the two together and inject the mice, guess what? The mouse dies. What does this mean? Right? So here's the data, right? I want you all to be the scientists now. What is your conclusion from this experiment? And feel free to use the chat. So someone said that the R strain picks up genetic information through transformation, right? DNA is transferred from the S strain to the R strain. Other guesses? Now, I know we're looking at this retrospectively, but imagine, put yourself in the scientist's shoes. DNA wasn't a big thing back then, right? They didn't know that DNA was the genetic information. So what is the conclusion from this data only? Right, nothing about this implies DNA, do you see, right? From this data, what can you conclude? Dead S strain plus living R strain equals living S strain, which makes the mouse dead. Exactly. You can isolate living S bacteria from this dead mouse. Someone said genetic information can alter strains. Other guesses? Yeah, and you pretty much all hit it on the nose, right? Something fishy is happening, right? Because you inject in dead S bacteria, the mouse dies, and then you look inside the bacteria and guess what? There's living S bacteria. Now, you could say, well, maybe the S bacteria were zombies and they were brought back to life. Or maybe the R bacteria were transformed into the S bacteria, right? And that's probably more parsimonious. That's probably more likely. And so this was called the transformation principle, right? Nothing implies DNA yet, right? There's no, isolation of DNA to see what exactly happened. But we do know that something happened, some transformation happened, and it transferred between the S to the R. The R strain, the R bacteria, got the killing trait from the dead S bacteria. Right? It's chemical. It's not something special to living things because this dead strain could also transfer that information. Does this make sense? If there are questions, please use the chat. Okay, so to set the stage, this means that there's something that can change traits. Now, someone asks, does transformation only happen in bacteria? I've heard a lot about it in experiments and stuff. And the question is no. Transformation can happen in other organisms as well. It was discovered in bacteria first, so good question. Okay, let's move on. So this is the conclusion. Some chemical can transform a cell because they have different traits, okay? And this is something that we see across the tree of life. So of course things have traits, right? Organisms have traits. I have black hair, I've got 5 fingers, right? I've got 2 eyes, a nose, a mouth, 2 ears. Those are all traits if you think about it. Like somehow that's encoded inside of me when I was just a zygote. And as I developed, I somehow got all these traits. And so our job as biologists and DNA thinkers, DNA philosophers, is to think about how that works. Bacteria, archaea, eukaryotes, you can see there's a whole range of organisms. And when something is this general, all these things have DNA, something's got to be up. And so, of course, now we don't really know that DNA is the genetic material yet. And so, Aristotle, you all heard of him, right? Fancy dude. He came up with this strange theory. Now, you all might be looking at this and then you might be thinking, well, this is kind of ridiculous, right? Aristotle thought, oh, well, the way that offspring got their parents' traits was from, some kind of temperature thing, if the heat overpowers the cold, then it's a boy, otherwise it's a girl, right? And the father forms, it's just ridiculous stuff, right? You can see, like, this was not based on experiment. This is just him philosophizing, thinking, right? Now, thinking is important, of course, but we also need to back it up with evidence. And so, throughout today, we're going to be looking at some of the data to try to figure out what makes heredity work. Now, of course, this is trash. This is not what happens. I'm putting this on YouTube, but this is not what happens. I want to emphasize that. Now, here was the question. Back then, people knew about DNA. People knew about proteins. You could isolate them. You could study them. But people didn't know what caused heredity. What is the thing that is passed from parent to offspring to make them look like each other, to have similar traits? Could proteins mediate heredity? Proteins have a 20-letter alphabet. Those of you who might have studied, a little bit of biochemistry know that proteins are made out of amino acids, and there's 20 typical amino acids that organisms use. How many letters are in DNA? Anyone know? 4. Exactly. There's 4 letters in DNA. So, right off the bat, proteins seem to be more complicated than DNA, right? They have many functions. Proteins, they could be antibodies, they give you immunity, they could, form structural polymers, they can catalyze reactions, they can be enzymes, they have many functions. DNA kind of just sits in the nucleus of the cell doing nothing. It's the proteins that are really the workforces, right? So people thought, well, maybe the proteins are, they're pretty complicated, and they can fold, right? So DNA has shape. Don't get me wrong; they can fold in all kinds of weird ways. But if you ask me what's more complicated shape-wise, I would have to answer proteins, right? Proteins fold in these really beautiful and elegant ways. And Colin and I are actually working on getting the structure of a protein. We'll see how that goes. But protein structure is super essential, right? They fold in these really beautiful ways. And there's all kinds of biocomputation you can do with these, all kinds of research going into the folding of proteins. DNA, you all said it, double helix, right? Oh, kind of boring. Yeah, Colin.
I also wanted to add like one fun fact that kind of, I don't know about DNA and proteins too, that kind of shows you how almost all DNA is somewhat useless in a way. But like DNA, 99% of it or 98% of it is non-coding, which means most of it is just regulatory. So only 2% of it makes all the proteins that your cells and all your body use, which is very interesting, I think.
Yeah, like 1 to 2%, and that's a wild number. I heard people call it like the dark matter of biology, you know? It's like the unknown exploring region, and most likely it's regulatory. People call it trash, junk DNA. Well, I don't think so, right? You asked me, 99% of the stuff is junk. I don't think so. Now, someone asked a really good question. Was RNA also a contender for who represents genetic material? I actually don't know. I'm sure some people believed it, but I don't think so because RNA is less stable than DNA. If you think about the stability of these molecules, in order for something to be passed down, it's got to live at least, a generation or longer than RNA does. RNA is very short-lived. It's very unstable. But good question though. So here's the fight, right? Proteins versus DNA. Who was the actual genetic material, and how do we figure it out? And to be honest, I'm looking at this slide and I'm thinking, I can understand why people thought proteins mediated heredity, right? Proteins, logically, it seems like they're more complicated. It seems like they could, tell the organism how to develop. That makes sense, right? But again, just thinking isn't enough. You gotta do experiments. And so here's my question to you all. How do you actually test what mediates heredity? And again, put yourself in the experimentalist's shoes. We got proteins versus DNA. How do you actually determine what mediates heredity? Think about it for a bit. It's not an easy question either. Because I feel like nowadays we all take it for granted. We all know that DNA is the genetic material. Someone said isolating DNA and proteins, comparing cells without them. Okay, so I like the idea of isolating. Whenever you do an experiment and you design an experiment, you want to isolate certain things you're interested in. So we're comparing proteins and DNA, we better be able to isolate proteins and DNA, right? Someone said experimentation on chromosomes. So chromosomes are DNA. And so I guess, yep, you can experiment on chromosomes to see if that will impact certain things. Find a way to tag the two compounds in question, then examine their activity in the cell. I like this as well, tag, right? So we'll get into that in just a very short bit. But the idea of labeling and tagging is really, really important in biology. Someone said crossbreeding. That's interesting because I'm actually curious. I'm trying to think about how crossbreeding would let you determine whether it's proteins or DNA. I could imagine crossbreeding and then isolating certain components, but that might be a little bit more difficult, but I think it might be doable. Isolate and compare DNA versus proteins, right? If there are more thoughts, put them in the chat. But for now, I'm going to move on. Phosphorus versus, oh, I see someone knows where we're going. So comparing alone DNA samples and analyzing their code per certain protein. I'm guessing that means percent proteins. So, and analyzing how they code certain proteins. Okay. Awesome. So let's move on from here. But now I want to actually show you an experiment that builds off of the first experiment that we were talking about. Remember that first experiment, the R and S, right? R is, benign. S kills the mice, sadly. You kill the S, mouse lives, right? This is what we saw before. You mix them together, mouse dies. Now, here's the new part. DNase heat-treated S and R together. What that means is it's basically this, but you add in DNase. What's DNase? Anyone knows? Type in the chat. Cuts DNA, exactly. Whenever you see the word ACE, A-S-E, that means enzyme for the most part. There are a few weird ones, but ACE means enzyme, and enzymes essentially catalyze reactions. They make reactions go faster. So DNase is an enzyme, a catalyst, that cuts DNA, breaks it down. What about protease? What does that do? What's a protease? Exactly, same thing, right? Breaks protein instead. So do you see how our experiment requires us to differentiate proteins in DNA, right? We need to be able to do something to the DNA but leave the protein alone, and vice versa. So that's how experimental design allows us to make these conclusions. Now, what do you observe? You observe that the DNA-treated S cells who are dead, combined with R, doesn't kill the mouse, but protease does kill the mouse. What does this mean? So focus on the last 3 rows here, right? If you just mix together dead S and R, mice dies. If you mix together heat-treated S and R with DNase, the mouse lives. But with protease, the mouse dies still. So do you see how something changed between these 2 rows? Right? These 2, nothing changed, but these 2 rows changed. So what are your conclusions? So someone said protease kills the mouse. That could be true, right? Maybe. But how would you test that, right? You could inject protease by itself, and it turns out the mouse won't die.
And cells have proteases in them.
Exactly. Our stomachs have proteases in them. So it's not the protease itself that kills the cell, but I like that guess, because without the control, you wouldn't be able to tell, right? So I like that comment, actually. DNA plays a critical role in transforming the dead R cells into living S cells. Exactly, right? The previous experiment, we couldn't say what exactly allowed this to happen. What mediated the transformation principle? Now we know it must be DNA. Do you all see why? Right? If we get rid of the DNA, then it's like nothing happened at all. Right? It's like the mouse didn't, So in this case, when we treat the S cells with DNase, we get rid of the DNA, the R cells no longer transform. They're no longer virulent. Right? But if we treat it with protease, we get rid of the protein, then the R cells are still transformed into killer machines and they kill the mouse. Right? Do you see how it's DNA that mediates transformation? I want to make sure that everyone's clear on this concept. Okay. Now, of course, this is only one small piece of the puzzle, right? I'm showing you a few experiments, a few key experiments, but again, this is not generalizable yet. Right now, we've only shown this in bacteria and mouse. But again, subsequent experiments show that this is true for pretty much any organism on Earth, all of life. It's generalizable. It's DNA that is passed down to change traits, to change, for example, the R into a killer machine. Any questions about this experiment? This is a landmark one. Now, people were still hesitant. Now, people were so ingrained. People still thought that proteins mediated heredity that people were still hesitant to accept that this conclusion was true. And so what do? You do more experiments to show this, right? So here's another experiment you can do. You can use viruses. Anyone know what these viruses are called? These little Mars rover looking things? What are these called? Yeah, exactly, right? These are called bacteriophages, or phage for short. And what bacteriophages do is they latch onto bacteria cells and they kill them. It's like a virus for bacteria. And they're bad news for the bacteria, as Winston said. They're bad news. Just like human viruses, like COVID-19, of course, is bad news for a human, bacteriophages are bad news for bacteria. And here's the experiment that I want to introduce to you all. And it was a study by Hershey and Chase. And someone alluded to this in the poll everywhere question just now. It's using sulfur versus phosphorus. Here's a key insight. And again, we need to be able to distinguish proteins in DNA. Previously, we used enzymes to cut one or the other. Now, we're using element labeling. We know from chemical studies, chemistry, that proteins have sulfur. And we know that DNA has phosphorus. So awesome, right? That way we can distinguish them. What's another prerequisite? So I told you 2, right? I told you that proteins have sulfur DNA has phosphorus. That's a given. What else has to be true for this to be an effective method to use element labeling to distinguish the two? What else must be true? Protein has sulfur, DNA has phosphorus. Is that sufficient for us to conclude? Yeah, exactly, right? Another important thing is that proteins don't have phosphorus and that DNA doesn't have sulfur. So do you see how now it's mutually exclusive? Right, now we can selectively label proteins with sulfur and selectively label DNA with the phosphorus, with the special phosphorus. They're radioactive, actually. So awesome. Now we have a way to distinguish the two. Again, we're using experiments to isolate the two components. And we know that phages infect bacteria to reproduce, just like other organisms reproduce. Phages can latch onto a bacteria cell, and then it does something to the cell to make it make more phage. So of course something's happening inside the cell to allow more viruses to be made. And their question is, what is the thing that phages transmit to bacteria to reproduce? Do you see how it's a similar question with transformation, right? Transformation was what allowed this trait to go from bacteria to bacteria. Bacteria hopping, essentially. Here, the question is, what makes information travel between phage to bacteria? Again, it's a transmission experiment, right? What's the chemical? Here's what you do. Label sulfur, label phosphorus, you then let the phage loose. At the very end, you notice that the cell itself has no labeled sulfur after it infects the cell. But the cell does have labeled phosphorus, meaning there's DNA inside the cell. What do you conclude from this experiment? In the chat. What does this mean? You see the phosphorus inside the cell, but not the sulfur. DNA is transferred and the proteins stayed. Exactly, right? Because the phosphorus was transferred from the virus into the bacteria, that must mean the DNA was transferred from the virus into the bacteria. Two experiments showing the same thing. DNA is what transforms things, is what mediates genetic information for at least phages and bacteria, according to these two, right? And this is more of a historical note, but I find it kind of interesting. In the actual paper, if you go to the very end, they say this protein probably has no function in the growth of intracellular phage. So they're saying proteins don't do this thing. It's the DNA that does. But notice how they phrase it. They say the DNA has some function. Further chemical inferences should not be drawn from the experiments presented. So you can see they're still hesitant, right? They're still like, DNA has some function, right? You can see how you can get, so ingrained by, I don't want to say, assumptions, but assumptions essentially, because people thought that proteins did it, but we know that DNA now does it. But back then they were still hesitant, right? It's got some function, but now we know it is the thing that transmits genetic information. Okay, let's move on. These are some famous pictures I'm putting on the slide. You'll see these everywhere if you take an AP Bio course, perhaps. This woman on the left is Rosalind Franklin. And this photo here is a photo, an indirect photo, I should say, of DNA. Now, you might be seeing, well, that looks nothing like the DNA we see in images and online, right? It looks kind of weird. It's got like breaks in it. This is what's called an X-ray diffraction image. So what Rosalind Franklin did was she took a DNA fiber, she shot X-rays at it, and the idea is that X-rays scatter off of the atoms inside of the DNA fiber. And depending on the phases of how the X-rays move past the DNA, there are certain dark spots and certain light spots that can show up on the screen behind it, which you can detect. And so this is a very famous photo. I don't want to get into the details of this, but you can, from the image here, you can deduce a lot about the DNA structure. You can deduce, the offset of the DNA strands, the width of the DNA, etc. But this X-ray diffraction image showed us that DNA was a double helix. That was a key, right? So again, it's an experiment that shows us that DNA is a double helix. And then Watson and Crick used this data to formulate their model of the double helix. So they have two strands wound around each other in a twisted ladder formation. And this is, they literally built these and they used their hands to build this model. And you can see it right here. So these are, historic moments in the discovery of the structure of DNA. So we've got structure and function. We know DNA transmits information and that DNA has this double helical structure, as we saw on the slide in the very beginning. Right? It's got this beautiful structure. Now, of course, this is an artist's rendition. This is not what DNA looks like. No way. We will get into what DNA looks like, and it's a lot of chemistry. So for those of you who might not have seen some organic chemistry before, don't be overwhelmed. I'll try to talk over it slowly. Now, this was a vocabulary term that was brought up earlier in the poll everywhere question. It's a nucleotide. Anyone know what a nucleotide is? Can someone tell me what a nucleotide is? We saw nucleic acid, right? Nucleic acids are things like DNA and also RNA, as we'll discuss. And they're found in the nucleus and they're acids. What's a nucleotide? Right, Jasmine says it's building blocks that form DNA. And also a base, right? So it does have a base right here. Other things. What's a nucleotide? Monomer of a nucleic acid. Exactly. So monomer means one piece, one Lego piece. And here's the idea where the Lego analogy comes back again, right? Imagine you have a Lego and you're trying to build, a mansion. You build that Lego mansion from single Lego pieces, right? So the Lego pieces are monomers and the mansion is a whole polymer. That's the idea. DNA is a polymer, right? It's a long strand of many, many Lego parts. And those Lego parts, those monomers, are called nucleotides. And there's three parts of a nucleotide. There's a phosphate, a pentose sugar, and a nitrogenous base. Now, of course, this is a very simplified diagram with just shapes. So we'll get into the chemistry very soon. But for starters, we have a phosphate group, a pentose sugar, and a nitrogenous base. Now, these terms might not mean anything to you, but let's see what they actually mean. Here's the phosphate. Right? What does phosphate sound like? We mentioned it already today. What does phosphate sound like? Phosphate sounds like phosphorus. Exactly. And guess what? There's phosphorus right there. That's what that experiment labeled. They labeled that phosphorus to detect the DNA. It's called a phosphate because it's got 4 oxygens around it. That's just terminology. A sugar. Who would have thought there's sugar in your DNA? Right? And that sugar just happens to be called deoxyribose. That's the first part of that DNA acronym. Right? Deoxyribonucleic acid. So there's a sugar in your DNA. And there's also a nitrogenous base. You can see there's nitrogens inside, right? And in this case, they're showing adenine. So this is a nitrogenous base. And here's what it looks like when you piece together those Lego pieces. So here's 3 adenines in a row, right? A. We say A because adenine starts with A. It's just the abbreviation we give it. And you can see how they link together, right? So for example, let me annotate. Here's a linkage, and there's a linkage. Right? Those of you who might have heard about the linkage, we call it a phosphodiester linkage. They're connected. That's basically it. So the nucleotides connect together to form the DNA polymer. So imagine this going on for millions of nucleotides. That's a chromosome. That's what might be a chromosome, right? So there's millions and millions of these all together. And you can see it alternates in a pattern, right? We have a phosphate followed by a sugar, then another phosphate followed by a sugar, another phosphate, sugar, and you keep going down, and you might keep going up, right? Potentially. So this is a trinucleotide. Your chromosomes might be millions of nucleotides long. Now, you can see there's not much variety here, right? To be honest, this is kind of a boring sequence. It's the same thing over and over again. What actually happens is there are different nitrogenous bases. And here are the 4 letters I was talking about, right? There's A, G, C, and T. You might have heard about that before, right? So adenine, guanine, cytosine, thymine, those names aren't too important, to be honest. I just call them A, G, C, and T. Okay?
There are more too that aren't just these that are used in other chemicals and stuff that are important.
Exactly, exactly. So these are kind of the canonical 4 base pairs. There's also something called U, which is an RNA. In fact, the Moderna mRNA vaccine actually uses a modified nucleotide with a modified nitrogenous base. So you can see, from an engineering perspective, as someone who's very into biochemistry, I'm thinking about all the ways you can modify these bases to have different properties. We have 4 bases now. Why not 8? That's Hashimoto DNA, right? People have, literally invented new DNAs by modifying the chemistry, and it's possible. People have done it.
And not to take too much time, but you can think about like information storage. Here's a purpose. You could, if you had 8 base pairs versus 4, how much more information could you store with that? Things like that, or how much more specific can you get? And some things to think about from an engineering point of view.
Yeah, exactly. There's also, for example, this might not be in the future, but it might, right? If we have 1s and 0s now on our computers, right? Binary. What if we had 4 letters, a DNA computer, or 8 letters even, 8-letter computer for information storage? That might be possible. If you modify a nitrogenous base, how can you be sure it will be transcribed or translated correctly? Yeah, that's a great question. That's a whole area of research. These artificial DNAs. Those of you who might have known about DNA before know that there are base pairs and you can pair them together. So just for instance, A pairs with T, they're friends, they like each other. G pairs with C, and they're also friends. And you bring up a really good point, Rohan. The new artificial base pairs that we make, we also make an artificial corresponding base pair to go with it. Yeah, so there's like, Z and S and, Z, all kinds of letters out there. But there are, new artificial base pairs that you can use. So very good question. And, not to ramble about this, but I'm rambling about this. But every single aspect of this basic structure can be modified, can be engineered, right? Who says you have to have a deoxyribose sugar? Right? There's all kinds of modifications. You can cyclize certain places. You can add sulfurs where the oxygens are. You can, in fact, there are, medicines that use that. You can add methyl groups here and there. You can even just disregard the whole sugar thing, and you can use, maybe amide bonds. Why not, to make it even more stable? Okay? So all kinds of biochemistry you can do to the DNA to engineer it, to modify it. Again, life is chemistry. It's atoms bonded together, and we know the rules of atoms and the bonding. So we can play with it. Right? That's the world of a bioengineer.
Yeah, and there's so much cool research going on in those kind of fields and stuff that we really recommend you look into. And when you're in college or even in high school, if you have the opportunity to do research in that, it's going to be super, super influential and very, very powerful stuff.
Exactly. And this is, this is the future. So anyways, let's move on. As I said, A is friends with T, G is friends with C, right? Very easy. I remember it because my name is Alan Tong, so AT. Very easy. You guys might have your own acronym, who knows? I remember Alan Tong, so AT. And the other 2 are G and C, they're friends. What do you observe? Why? Why do these happen? Like why are A and T friends? Why not A and C or A and G?
Look for the differences in the 2.
Right. Compare and contrast, right? These are different chemical structures. And I realize that, some of you might not be familiar with, these chemical structures, but just big picture wise, right? So here's an A, here's a G, here's a C, here's a T. Big picture wise, what do you notice? Rohan said same width. What do you mean by that, Rohan? Big plus small. Exactly, right? 2 rings, 1 ring. 2 rings, 1 ring. Perfect, right? Because of the same size, DNA has a constant length. I believe it's about 2 nanometers, right? If you do big and big, that might, have some chunky parts in the DNA. It's not as well structured. Very good. And those of you who might want to go more in detail realize that there are hydrogen bonds between these that can also facilitate the base pairing. Exactly. So amino set depends on the hydrogen bond. There are 2 here and there are 3 here. Very good. For those of you who know about hydrogen bonds, awesome. Those of you who don't worry about it. It's just, attraction, essentially. So these two attract together. A binds with T very nicely. G binds with C very nicely. Right? Think magnets, although not really magnets. It's more charged, but anyways. And now we have our base pair rules down, and here's the big picture. So here we just showed the nitrogenous base by itself. Here's everything all put together, right? Here's the nitrogenous base, here's the sugar, and here's the phosphate. Nitrogenous base, sugar, phosphate, right? Base, sugar, phosphate, base, sugar, phosphate. You can see there's a pattern, right? Schrodinger called this the aperiodic crystal. So it's a crystal, but it's also aperiodic. It's not like a regular crystal, which is really structured and periodic, very regular, right? DNA, you can modify these sequences. You can do TG, as I have here, or you can do AC or AGC, or the million sequences inside of your chromosomes, right? So these are all things you can do with DNA. But again, we still haven't answered the question of how DNA encodes traits, right? We've kind of talked about 2 parts of the story. We talked about how we discover DNA's function and the structure of DNA, but what's the thing that connects it to? And I'm sorry, Colin, I saw you unmuted.
I was just going to say, if you want to answer Rohan's question in the chat really quick for everyone.
Thank you. Why don't the negative phosphate groups resist the pairing of DNA strands? That's a really good question, right? So you can see here, phosphates are negative, as we saw, because they're acids, right? They release the protons. This phosphate's negative, this phosphate's negative. Why is it that they can come together, right? That seems to be, counterintuitive, because negative should repel negative. And you're exactly right, it does. But there are also counter ions. And so inside of the cell, I believe there are magnesium ions that you can coordinate to the phosphoruses. Magnesium ions are magnesium 2+, and the 2+ attracts the phosphate negatives and almost masks that negative charge. And so it's almost like it's neutralized and you won't be able to see the charges. They are still there, but it's to a lesser extent. And it just so happens that the millions of base pairs between the 2 strands overcomes that repulsive force. But that's a really good question. I can see you're really thinking, at a chemical perspective. Why don't these charges attract or repel? They do repel, but it just so happens that the hydrogen bonds attract more than it repels. Really good question. Awesome. And these 2, if you extend this out for millions and millions of base pairs, you get your double helix twists as well. And here you can see that twist, right? Let me check on time. Oh my goodness, we have 5 minutes left. Wow, I got a lot more planned. No worries, I'll just leave it the next time. But here's the structure, right? Here's the beautiful thing we all know and love called DNA. You can see how it's composed of all these elements, phosphorus being the thing that's different from, proteins. And you can see how it's also 2 rings, 1 ring, right? 2 rings, 1 ring. A pairs with T, G pairs with C. Why does it twist? Anyone know? Why not just a straight ladder? Why does it twist? Any guesses? If you think about what's different between the regular ladder and the twisting form, or maybe give it another few seconds. Compact, right? That's one thing. It makes it more compact, right? The nucleus is a pretty small thing. You can't even see it with your eyes. So yes, it does make it more compact for sure. And also more importantly, okay, so Eileen says to save space, so compact, and also to protect the bases. Yes, it's to protect the base pairs from what? From water. It turns out your cells are chock full of water. And it turns out that the base pairs don't like water that much. So if it twists, it can hide the base pairs from water. So interesting tidbit right there. But again, we haven't connected it to traits. We see the structure, we know the function. What's the connection? And I think people have alluded to this. And here's just the image of DNA coiling up into your chromosomes. You might have seen chromosomes before. All right. There are these chromosomes are just really small noodly DNAs inside your nuclei. And you've got 23 pairs of them, right? 23 pairs. 23 from your mom, 23 from your dad. And you can see that there are different types of chromosomes. Some are big, some are small. These last two are called your sex chromosomes. XX is biologically female, XY is biologically male. But okay, DNA is passed from your parent to offspring, right? You get half your DNA from your dad, half from your mom. But again, we still haven't answered the question of why does DNA encode traits? Let me check on time. Oh gosh, we got 2 minutes left. I want to end on this note, right? We've got a whole sequence, a whole genome. That's the term we call it. The sequence is called a genome. But what is it that codes for the traits? And here's one idea. Oh, okay, I have a whole other question for this. How does the DNA sequence lead to traits? Wow, I should really study this before joining the call.
This is a good place to end for next week too, where it's some of your thoughts, and then we'll tell you more starting next week before we get into the other information.
Good idea, good idea. So let's ponder over this over the week, and I will see you all then.
And
so
one last thing, try to show up maybe like 5 minutes early, if you are able to, just so we can get started right at 6, and not really have to do anyway, because there's a lot of information to go through, and we really want to teach you all everything from the little bits, because we're going to really build on these subjects, and we don't want to leave anything out of it.
Awesome. Thanks, Colin. All right, we already got some answers. But yes, so you're all free to go. I will stay on maybe for a few minutes later to answer any questions you have or to look at these responses. I know, Colin, you have something in a bit, so.
Yeah, I have to head out, so thank you all. But yep. Alan knows everything. He's all seeing. All next week. And if you have any questions, email us too.
Right. Awesome. Thank you all for joining. It was really great having you all. Thank you for being so engaging, using the chat, doing the poll everywhere. And yeah, I'll see you next week.
All right, cool. Well,
awesome.
All right, Alan.
All right.
Continue the recording, and I can edit it. And
I will send you. Let me stop recording actually.